Ever wonder how scientists can pull off super precise surgery on something as tiny as DNA? Well, today we're diving into an incredible technology that does exactly that. And the wildest part? The whole idea came from a tiny virus with a very, very clever plan. So let's see how we get from a natural wonder to a revolutionary tool in the lab.
Here's what we're going to cover. First, we'll talk about the challenge of editing DNA. Then, we'll uncover the secret cut and paste trick that a virus uses. After that, we'll get right inside the gateway system, see how it can actually run in reverse, and finally, we'll talk about why this is such a huge deal for science today.
Okay, so first up, smarter DNA editing. We've got to start with the fundamental problem here, and that is the challenge of precision.
I mean, this is really the million-dollar question in genetics, right? Working with DNA is kind of like trying to edit a massive 1,000-volume encyclopedia. You can't just go in there with a Sharpie and cross things out. You need a tool that can find the exact word on the right page in the right book and replace it perfectly, every single time. That's the insane level of precision we're talking about.
And it turns out, nature already figured this out ages ago. The inspiration came from a virus, which gave us what is basically nature's ultimate cut-and-paste trick.
The entire foundation for this technology comes from this ancient battle between the phage lambda virus and its favorite snack, the E. coli bacterium. See, the virus needed a way to sneak its own genetic code into the bacterium's DNA, and it developed this incredibly elegant method to do it without making a mess.
Okay, so the fancy scientific name for this viral trick is site-specific recombination. And the part you really got to focus on is site-specific. This is not a random process. The virus doesn't just jam its DNA in anywhere and hope for the best. Nope. It exchanges genetic material at very precise, predetermined locations. It's smart. This ensures the virus doesn't accidentally break any of the bacterium's essential machinery.
All right, so let's peek under the hood of the gateway system and look at the actual locks and keys of DNA that make all of this possible.
So here's the secret sauce. Both the virus and the bacterium have these unique DNA sequences that basically act like little docking stations. On the virus's DNA, you've got a site called at P, and on the bacterial DNA, you've got one called at B. Think of them as the specific locks that are literally designed to find and recognize each other.
And here's the play-by-play. First, those at TP and at B sites find each other and line up perfectly. Then, a couple of enzymes, one from the virus and one from the bacterium, they come in and work together like molecular keys. They kick off the reaction, snip the DNA, and then seamlessly stitch the viral code right into the host's chromosome. And in doing so, they create two brand new hybrid sites.
And this just shows you how clean that change is. You start with at P and at B. After the reaction, poof, they're gone, completely consumed in the process. And in their place, you have two totally new sites, at L and at R, that are now flanking that newly inserted piece of DNA. It is a perfect one-for-one swap.
Now this is where you can actually see the proof in the pudding. The original at TP site is super tiny, just 25 nucleotides, or letters of DNA code, long. But after it combines with the much bigger at P site, look at that! The new sites have completely different lengths. Now for a scientist in the lab, this size change is everything. It's a perfect fingerprint. It's an easy way to run a test and get a bright flashing sign that says, hey, the swap worked exactly like it was supposed to.
But here's where it gets even cooler. This whole process isn't a one-way street. The virus also figured out a way to reverse everything when it needs to make a quick exit. This next part is all about putting the pieces back where they started.
So let's say the virus needs to pop its DNA back out, maybe to go make more copies of itself and infect new cells. Well, it has an escape plan. It calls on another tool from its toolkit. This requires a third enzyme, a protein called IS. This is the special key that unlocks the reverse reaction.
And this table just sums it all up perfectly. You've got the virus bringing the at P site and the int and axis enzymes. The bacterium brings the at B site and a helper enzyme, IHF. Now, notice how int and IHF are used for both putting the DNA in and taking it out. But look at X. That's the crucial switch. It is only needed for the excision, or the removal process. It's the get me out of here enzyme.
Okay, so a sneaky virus, a few enzymes, what's the big deal? Why does any of this matter to us? Well, this is where we go from a cool trick in nature to an absolutely essential tool in the modern lab.
So this is the big aha moment. Scientists looked at this incredibly elegant, efficient, and reversible system and thought, wait a minute,
We can use that. By basically hijacking nature's own cut-and-paste tool for the lab, they created the powerful and reliable system we now call gateway cloning technology.
This is a game changer because it made advanced genetic engineering accessible to way more people. Before technologies like this, editing DNA could be messy and unpredictable. Gateway gives researchers a standardized kit that delivers predictable results, which means a scientist anywhere in the world can easily stitch together multiple pieces of DNA with confidence. And now, this is even backed up by amazing software like Benchling that lets you plan out these complex reactions on a computer before you even touch a test tube.
And get this, for academic researchers, students and scientists at universities, the price tag for access to these incredibly sophisticated cloud-based tools is often zero, nothing. This just demolishes a huge barrier, allowing people everywhere to design cutting-edge experiments using this powerful technology.
And that just leaves us with this final, really incredible thought. This entire revolutionary technology was basically reverse engineered from a simple virus fighting a bacterium. It just makes you wonder, doesn't it? What other amazing molecular machines, what other undiscovered toolkits are just waiting out there for us to find in the vast, unseen world of nature?